All right, welcome to our webinar, Unveiling Environmental Microbial Mysteries, Harnessing the Phenotypic Approach. My name is Kasia Proctor, and I'll be your moderator today. Just a few housekeeping items. Your lines have been muted, so if you have any questions or problems, please type in to the Q&A window. Our speaker today is Dr. Enrico Tatti. Enrico is a global field application scientist at Biolog. He's based out of Italy. He obtained his PhD in soil microbiology from University of Florence with a thesis on the effects of agriculture practices in soil microbial communities in the Chianti area. Welcome, Enrico. Take it away.
Thank you very much, Kasia, for the introduction, and thank you, everyone, and of course, good afternoon. Thanks for being here. And we're gonna spend the next 30 minutes together, and we're gonna talk about microbial communities and the best approach to analyze them. So when it comes to the analysis of microbial communities, I like to start with a question, or if you want, a concern that I have. And the question is metagenomic really the only way that we have to define microbial communities? So we know that if we look at literature that most of the papers that are published analyzing the microbial community from soil or from water, from sediments, wastewater, any type of environment, they generally rely to molecular methods that entails the extraction of DNA or RNA or both, their purification, their amplification, and then their sequencing. And this sequencing typically rely on targeting 16S RNA. And this is you can see from the slides, the typical data outline that you can get from such an experiment. This is actually a very nice paper from Chang et al. Published in 2021 on applied soil ecology. And it shows that after an analysis done with targeted metagenomics on a soil sample, for example, for this particular case, we end up having a list of different microbial groups. And this can be done and obtained at various different phylogenetic resolutions. This list also can expand and give a little bit more of extra information as the relative abundance of each group. And this information can be used to appreciate changes of the community over time or in response to a certain treatment. In this particular case, the experiment was done after a treatment and repeated for 2 consecutive summers. So we can appreciate in this type of data sets how the different microbial groups change over time and how they remodulate themselves and how the community substantially respond to different environmental factors. But in an analysis like this, where we have access to the different groups, we actually have no information on function whatsoever. So we see that these microbes are there. We may also know that they are active if we analyze their RNA, but we cannot relate any of these groups to a particular gain or loss of function. And certainly we can expand using molecular methods the analysis from targeted metagenomic to untargeted metagenomics and actually sequence the entire pool of functional gene there is in the microbial community in the sample under analysis. And doing this, we have to enlarge a lot our sequencing effort, and there's the need of great computational power to process all the data. And we can then get a list of all those genes that are present in the sample if we do so. But anyway, even if we select to go down this path, we may still miss what the community may be actually capable of doing. And this is because the relationship between the presence of a gene and its function in the environment is not linear. In other words, the genotype-phenotype link is quite complex. And this is very true for single strain, but it's even more true if we think a complex microbial community. So we may get a list of the pool of genes that are in that sample, but we are not sure that functions are actually performed by the community because most of these functions may be related to the environment or to regulation. And also, even if we analyze one gene one by after another, we know now that it is true that one gene may be related to one function, but it is also true that one gene may be related to two or multiple functions. Some genes are heavily regulated, some other are silent. And also looking at the genes as a list of possible putative function or actual function, we are sort of losing the synergies that this gene may have across each other, and also the emerging properties that may come in, by the interaction of this pool of genes. So it is definitely a great level of information that we can get from metagenomics, but it's not the only information that we need. Actually, a method that would provide an overview of what the community can actually do, what are the functions that the community can perform in that given environment, would be extremely useful. And that's exactly what the biolog technology does. So the biolog technology, first of all, is a cellular analysis. So it will work on cells that are alive, while we know that metagenomic methods, they rely on the extraction of DNA and RNA, so killing the cells and then extracting and sequencing these macromolecules. But in case of the biolog technology, it will work on the entire cell of the entire community. And it's a rather simple method that is outlined in these slides. Actually, what you can see here is a typical 96 well plate, one similar to one that you may have already seen in your lab. And the peculiarity of this plate is that each different well, apart from a negative control, contains a compound that is meant to test for a particular function that the community may have. So, for example, well may test for the capability of metabolizing amino acids. Another well may test against the capability of metabolizing sugar, and so on. And all that the user has to do is to extract the community, the cells from the environmental matrix, and add it in a standardized way to the well, meaning that trying to add the same amount of cell in each well. That's, of course, very important. So the cells can be added, and the entire community will be added to the plate, as I said. And in the plate also, the cell will find a redox dye, which is an indicator for metabolic activity. And actually, this redox dye will be able to produce a color in the well if the microbial community has that function active, if it's metabolically active in that particular functionality. If it's not, it will leave there will be no color formation in the well. And this color, of course, can be quantified. So at the end, what we will have is with this quite straightforward experiment, we will have a functional readout or a metabolic fingerprint of what the entire community can do. And this can be quantified, and we will see in the next few slides how and what type of data we can get out of it. So if we use this technology, which is quite straightforward, as I said, we will basically see what the microbes can eat or what they can catabolize. And actually, during this metabolic reaction, if they're happening, of course, what will happen is that molecules like NADP will be reduced by an NADPH instead will be produced by the cells. And this molecule is what will interact with the redox dye that initially is in an oxidized state and forming no color, but the more electron we get from NADPH, the more reduced it will get and the more color will form. So the color that it will be possible to see over time after the incubation of the microbial communities in the biolog plate can also be quantified. So using this simple technology, but very informing, we can answer essentially three very important questions. First of all, what are the functional traits of the microbial community? Then how much active are these functional traits? Because we can quantify the dye color production, and then also how fast or how efficient are these functions, because we can measure the color formation over time and access kinetic information. We'll see this a little bit more in details in the next few slides. So after saying that, what are the difference and the possible synergy with metagenomics? This method is not better or worse than metagenomics, but is different and provide a different angle. And we think that can provide also a great synergy when used together with metagenomics. So we know that metagenomics is a method that works in any sample with any microbial community, so soil, water, wastewater, also host-associated microbiota, so gut microbiota, and doesn't require the cells to be alive. So the sample can be collected and then placed flash freeze, and then analyzed at later stage. And then it will provide, as we have seen before, information on the community composition and also how this composition and structure of the community can change over time or in response of a treatment. As we have seen, this will mean that we're going to break down the community in different microbial groups, and we're going to see how the community is composed by these different microbial group and what's their relative abundance. This is a reductionist approach, because instead of looking at the community as a whole, it will try to break it down in different parts. And also, as we said, it's going to miss the information about the functionality. Instead, the biolog approach, which is also known as community-level physiological profiling, also can work on any sample, so soil, water, sediments, and host-associated microbiota, but it will require, since it's a cellular analysis, it will require cells to be alive and metabolically active. And instead of showing community composition, it will show community functionality and how these functionalities or metabolic traits will change over time in response to a treatment. Since this will take a look at the entire community, is a more holistic approach. It will look at the functionality of the entire community instead of breaking it down in different members. So for this reason, the two methods can be really complementary. And these slides is to show and explain that. So we know now that if we have different communities, say like community 1 and community 2, they may be characterized and by different members, like the one, a different letter means a different microbial groups. But what matters is what are the functions that you can see outlined here on the left. So the capability of, for example, of degrading some amino acids or protein or peptides and so on. It is important to add this information because we know that a community, despite on being different in their composition, they can be functionally redundant. So they may have the same functionality. And with the biolog method, we can see if that's true, if different community have same or different functionality. And even if they have different functionality, we can differentiate even more by the efficiency that they may have in each of these functional traits. And we can do, because we can quantify the amount of redox dye, the amount of metabolic activity, and also analyze it over time for kinetic information. So again, the union of metagenomics and biolog technology can provide a more complete picture for the analysis of microbial community. Let's have a look now at what are the main tools for the analysis of microbial communities when the biolog technology is used. The first one that I'm gonna talk about is EcoPlate, and I'm sure many of you have heard about EcoPlates. It's a great classic. It's a 96 well plate that actually has a suite of 31 carbon sources that are environmentally relevant. These carbon sources are particularly interesting because they are not simple sugars, but they are more complex sugar. We will see in the next slides a little bit more in details. And another peculiarity of the plate is these 31 carbon sources are repeated 3 times. So the user actually can decide the design of the experiment, if they want to do replicates, or if they want to test different samples on the same place. So it gives also flexibility in terms of the experimental design. And the compounds, of course, are really important. There are, as I said, the carbon sources are more complex, and they be divided in functional classes. For example, carboxylic acid, carbohydrates, amines, phenols, polymers, and amino acid. You can see here that some carbohydrates are like plant derivatives, so more complex, like cellulobiose or xylose. And so the plate can, the echo plate can provide information on the functional activity, and you can see here also granularity about the actual function in various level of details, a larger level for functional classes or more high resolution on specific functional that are dictated by the carbon source that are in each well. So carbon source is the main feature for echo plates, and echo plates is actually one of the options that the user have if they want to analyze microbial communities with the bio approach and the phenotypic approach. Echo is one of them. There are actually other plates, and I would like to point out, for example, the FF plate, which is a very interesting plate for those that are interested in analyzing fungal communities. Then we have an anaerobic plate, a specialized anaerobic plate, very good to analyze anaerobic communities, so host-associated or human microbiome, but also anaerobic soil, anaerobic sediments, or wastewater. And then also the PM plates. And the PM plates are, I think, very, very interesting for the reason that allow the user to go beyond the carbon source functionality and can test, for example, for functionality on nitrogen by the microbial communities, and this can be really relevant in soil or on sulfur and phosphorus, very important macronutrients for many environments. So the plates are actually one of the two components of the BioLoc technology. The other component is the Odin platform. The Odin instrument is capable, is a really powerful instrument that is capable of incubating multiple plates, up to 50 of them, and to read them automatically at every 20 minutes. So the user can add the multiple plates in the Odin platform, and they will walk away, and they'll be read automatically. And Odin is a spectrophotometer, so we will be able to read the different wavelengths. It can read the wavelengths of 590s or 490s, which is great for metabolic activity and reading redox dye. It can also read at 750 nanometers as multi-filter capability, and in that case focusing on the growth and the biomass growth that is happening on the plate. So very, very powerful instrumentation that can really increase the resolution of the analysis. And this is because, as I said, Odin is an automatic reader. It will read automatically the plate every 20 minutes, and it will plot the signal intensity, either dye intensity or biomass amount, or over time, providing for each of the test in each of the plate that is incubated a kinetic curve. And the kinetic curve, as you can see, has a lot of data into it that can be easily extracted by the data analysis software that is present in Odin. Data important like area under the curve, or slope of the reaction, or the inflection time and the lag time. These data can be all used to characterize and get more information about the functional capabilities of the community. So especially if you have to do a comparison between condition A and condition B, so the community before and after a treatment or after a certain time, Odin can really be useful for that, because it can collect high resolution kinetic data and compare these data sets and highlight even the smallest difference for a very high resolution analysis of the communities. So let's see now that we have seen the different plates and the Odin instrument, some methodological aspects that are related to the biolog phenotypic approach for microbial community analysis. So we have seen, as I said in the first few slides, the process of setting up the plate is quite simple, and you just have to extract the biomass from the environmental matrix and then add it in a standardized way to the plate, and then incubate over time. There will be color generation or growth, and that can be measured using the Odin instrument. Anyway, there are three key points that need to be kept in mind, even if the experimental setup is very simple. And one is sample preparation. Sample preparation requires that you get a representative pool of cells that is reflecting the actual community in the cell. So for soil, the cells need to be extracted carefully. Also from water, often they need to be filtered or centrifuged to reach a certain amount. That is good, but most importantly, the cells, they need to be kept active, metabolically active. So no sudden change of temperature, no physiochemical stress. Then generally, the cells are diluted to make sure to add the same amount of cell in each well and minimize heterogeneity. The level of dilution will change from sample to sample. It's never the same, so it may need to be found empirically, but it's important that the same inoculum is used if different samples are compared between each other. So inoculum density is definitely a very important factor. And the third important factor to keep in mind is the incubation. First of all, the temperature. The temperature has to be relevant to the environment that is under analysis. So if an analyzing compost that is at 45 degrees, it makes sense to incubate the plates at 45 degrees. And very, very important is the incubation length. And these slides also show why. If the cells are added to the biolog plates and they're left there for too long before the data is collected, it may be possible that some of the cells that are in the wells, they start dying. This is because of the time that has passed, also because they are in artificial condition. So dying, they may release nutrients, and these nutrients, they may be uptaken by the surviving cells and used, producing a background color that is the same in each well. And this is something that is not ideal, because there won't be any difference between positive and negative. What is ideal is to read and analyze the plate in the so-called midpoint pattern, where you actually have a difference between positive and negative, and all the analysis can be done. So it's important to read and analyze the plate at the right time. And Odin, being an automatic reader, can really help in this. Odin will collect data for as long as the user want, 1 day, 2 days, 3 days, 10 days, and then it will be very easy then to retrace back and go and analyze the plates at the midpoint pattern and get meaningful results. Also, Odin is going to be really helpful in the further data analysis pipeline. The user can decide if you want to analyze on single points or on all the spectra of the experiment. So using also the kinetic. This is all possible because of the data analysis software that comes with Odin. You can decide easily if you want to use a certain parameter, either under the curve, the slope, or the inflection time. It can really help then to standardize and normalize the results and channel then these data sets into univariate or multivariate data analysis. This is because, as I said, the data analysis suite that is in Odin is really flexible and also really powerful. It can prepare data that is ready for export and use in software of your choice. And then the user can decide if you want to use, for example, univariate indexes like Simpson or Shannon or evenness index, or channel all the data sets into multivariate data analysis like PCA or MDS or cluster analysis. So this is really helpful and can really speed up and add great values to the functional analysis done with the biolog approach. Okay, so in this last part of the presentation and time that we have together, I would like to talk about some very good paper that shows how the technology has been used. I have selected 3 papers for this purpose, 2 that outline a typical use of the technology, and 1 there is a more non-standard use. But before that, I want to mention to you 3 great classic papers that are really good for anyone that is approaching the technology for the first time. They provide great information on how to set up the experiment correctly, how to analyze the data, and how to interpret the data and avoid any overstatement or pitfall. So these are really good, and you can find them easily also in our paper repository. We have a publication database in our webpage that I suggest you to explore. There's more than 5,000 publications where biolog technology was used, and there's some filter that you can apply. So for example, the plate type or the type of application that can really help to find something relevant to your research focus in a very short time. So I mentioned I wanted to present you 3 good papers. The first one is a paper from a Polish group led by Maciek, and it is a very relevant paper for sustainable agriculture. In this particular experiment, the scientists, they had a biofertilizer made by 3 strains of Bacillus and Pennibacillus, and they added this biofertilizer to the soil, and they wanted to use different approaches to see how the resident soil microbiota would respond, focusing on both bacteria and fungi. And to do so, they decided to follow 3 different approaches. One measuring enzymatic activity, another one measuring the community composition and structure with focused targeted metagenomics on 16S and ITS, and also TRFLP, and then the functionality with the biolog plates. And in this regard, using the biolog plates, they focus on 2 plates that I mentioned also early. One is the echo plate for bacteria functional analysis, and the other one is the FF for filamentous fungi functional analysis. And what they did was not just to focus on the metabolic activity, so just on measuring the dye color formation, but they also measure bacterial and fungal growth. So they use these dual wavelength readings. This is something that you can do with the Odin instrument at 500 nandies for metabolic activity and 750 for biomass. And then they calculated this ratio between metabolism and biomass, a ratio that is an excellent proxy for the level of stress of the community. If the ratio is high, so there's more metabolic activity than growth, it means that the microbial community are stressed and they cannot convert energy into biomass. Vice versa, if this ratio is low, it means that the metabolic activity is then channeled over to growth and biomass. And this is, of course, a good proxy for a less stressed microbial community. So let's have a look at the results for both fungi and bacteria. In these slides is a little bit more in details. So on top you see bacteria, and on the bottom you see fungi. And it is important to state that this experiment was repeated for a different amount of a biofertilizer, but also over time. So there's a time gradient from left to right. And it's interesting because you can see that the blue color is associated to a lower amount of the lower value of the ratio, so less stress, while warmer color is associated to a higher amount of stress. And there are some functionality that have been broken down by different functional classes that have been affected. Some of them, they have higher stress, but the majority of them, they have lower stress in the bacterial community particularly, and there's also legacy effects that is possible to see on particular functional guilds, the carboxylic acid that is kept over time. So really high details and real great granularity of the analysis that can be then complemented by NGS sequencing that can show, for example, an increase of the total diversity and how this diversity change and if it's kept over time. Interestingly, the fungal community didn't show any trend after the biofertilizer addition, and this is somehow is expected and also confirmed by NGS data. So an excellent example here on how these two methods can be used in conjunction and give great outcome and great information. The second paper instead is from a Chinese group led by Li, and is published on Chemosphere early this year in 2023. In this case, the users, again, they inoculated something in the soil. In this particular experiment, they took endophyte extracted from plant and they inoculated them into the rhizosphere of a cadmium removing plant, Phytolacca acinosa. And they then used EcoPlate to study how the resident microbial community would respond to this addition. It is known that the addition of this endophyte to the rhizosphere would increase the phytoremediation ability of the plant, but also it was possible to see using the EcoPlate how the resident microbial community would respond. And in this case, the average color well development parameter was used, is something that can be easily calculated on each of the biolog plates used for microbial community analysis, and it's an index of activity. This index of activity was also broken down into the different functional classes and followed over time for the different treatment, and gave it a great granularity on the specific response of the community following this addition. So really EcoPlate in this case, used also as a standalone method, was providing and working and functioning as a valid quality control tool to see and understand the response of the resident community after the addition of this bioproduct. But this can be repeated after a treatment or after time, still providing the same level of information. The last paper that I want to present is the one that has from Flynn et al., published in 2017 on Frontiers in Microbiology. And this paper is particularly interesting for the reason that it's been used in a non-standard way. So EcoPlate in this case were used as mini bioreactor. So different soil samples were added in different EcoPlate, and each well will represent a different growing condition. So the cells from the environment will added in the plate, and then depending on the carbon source plate, the carbon source would push for the change in the modification of the community towards a less diverse community or coming up different member of it. And this member could be then taken, from each well and plated and then isolated to foster the isolation of previously not isolated species. And these species then could be used further for biotechnological application. For example, the isolates could be characterized with functionally with a biolog plate, and then separated with a simple multivariate data analysis like this PCA here. And in the light of the possible device of a biofertilizer or a bioproduct that can be applied of soil, the most functionally different could be logically placed together in order to add more diverse functional traits into the soil and provide the soil with additional and different metabolic activity. So this is a nice example on how the technology was used beyond the standard protocol and how flexible also the biotechnology, the biolog technology can also be. Okay, so we got to the end. I just want to close with some final remarks. analyzing microbial community with biolog plates, as you have seen, is quite easy to perform. There are some key points to keep in mind, but it's anyway quite straightforward and also low cost. The data analysis, thanks to the Odin instrument, is easy approachable and can provide a lot of information, including kinetic information and rates about this reaction, not only the presence of certain metabolic capability, but also the rates and the efficiency. And is certainly a great complementary data sets to have and to add to sequencing and vice versa. And also as different type of application, you can target anaerobic bacteria, you can target fungi, you can target the whole community, you can target nitrogen, phosphorus, and sulfur functionality. So very also flexible for additional application. And last, Odin also, the instrument makes your life easier in terms of the possibility of incubating a larger amount of plate, keeping them at the desired temperature, not perturb and disturb the experiment, reading them at predictable intervals, so not missing any important point, and help in the organization and the analysis of the data. So I got to the end. Thank you very much for this time with me, and of course let us know if you have any question.
All right, thank you, Enrico. So as noted earlier, if you have questions, please type them into the Q&A window. And for those of you that have to run, I know we're running a little long here. We are recording this session, so we will be sending out the link to the recording if you wanted to hear kind of the last few questions or something. Let's see, we have a question coming here. Do you have a rough estimate of what percentage of the soil microbiota can be detected by biolog?
Oh yeah, excellent question. Thanks for asking. So biolog technology in the past has been considered like a cultivation technique, but actually this is not the case, because you are adding a complex community in there, and there will be certainly some selection on some members that won't be able to perform or growth, but it can still provide up more than the plating count or isolation technique percentage of the microbe. So it still give a very reliable picture of what are the active species, the one that are most likely contributing to the ecological function that are happening in the sample under analysis.
Great. The second question is about the dye. Is the tetrazolium redox dye used in eco plates different from that used in the FF plates developed for the fungal identification? And then what about the Gen 3 and then AN plates?
Yes, yeah, very good question also as well. We get of course this question several times because of course redox dye is very important, and yes, so the eco plate and the FF plate, they have different dyes, as you could probably see from the picture. The eco plate dye is gives out a purple color, and the FF dye is more a salmon red color. So yes, they are different because they're modification on the tetrazolium ring that to modulate their compatibility and with the, for example, fungi. So also AN plate has doesn't have really a different dye, is more the condition on where you run the AN plate. The Gen 3 plate also have a similar dye to the one in the AN and the Eco plate. So and also has the feature that you can avoid adding the dye, so focusing only on growth. This is also a feature that that is present for PM plates in case the user wants to focus only on growth instead of metabolism. So there's different possibilities, and yes, the redox dyes are different and they have different efficiency depending on what's your target, fungi or bacteria.
Okay, thanks, Enrico. There is one question here that I might be able to answer. It says, thank you so much. Where can we find description of methodology? You're definitely welcome to visit our website, and there's a lot more information there. And publications, as Enrico stated. And if you have further questions, please feel free to reach out to us with the email here on the screen at the info@biolog.com. There is a request, Enrico, to show the title of the third paper one more time, the one that was used in the atypical approach.
Sure thing.
There you go. Thank you.
So we also have a webinar for this. It was done a few years ago, but the authors gave a webinar for us regarding this paper. So a very good and interesting one for sure.
Yeah, so if you're interested in that particular one, we can probably look up the link to that recording and send to you. There's another question. Is it required to keep these plates in incubation in darkness?
Yes, I would recommend to keep them in darkness. So there's the redox dye is light sensitive, not to the neon light sense for sure, but to natural light. So I would recommend to keep them in incubation and in the dark if possible.
Okay. There's a question. Does the same apply to microstation or just Odin?
It doesn't apply, unfortunately, to microstation because a microstation is it can read only one plate per time, and it also doesn't incubate the plate. So you would have to take the plate out of the incubator and read it at certain times. Of course, if you have a large number of plates, that's not ideal. And also, if you want to have the same resolution that you have with Odin, you would have to read very often. Again, not ideal to do manually. Also not ideal for the sample that is moved, from the incubator, change the temperature, and then put it back. So the microstation can be helpful, but it doesn't get really the same level of analysis as Odin.
Okay. There is a question. Is it possible to put biolog plates in the centrifuge to spin down microbes?
Yes, it is possible, depending on what's your goal, right? If it's the end of the incubation and you want to take the supernatant for whatever reason, maybe you want to analyze the metabolites in the supernatant, or you want to do any type of test on the supernatant, yes, the test is done, no harm done to the cells. But as I said earlier, one key point is not to stress the cells. So you have to keep the cell alive. And sometimes some cells, if they are manipulated too much, if they are centrifuged, if they are subjected to physical chemical stress, they may lose their metabolic fiber. So I would recommend to do that only at the end of the incubation, if possible.
Okay, thank you. There is a question here. Would you like us to share published material where we used biolog? Absolutely, I'll take the answer here. So please, I have your name here. I can reach out. We are always open to customers sharing their stories. As you saw Enrico present, there is many of them on our website already. But we'd love to hear from you and how you used it. So thank you for reaching out. Another question is, can the system be used to phenotype archaea microorganisms?
I was discussing this not long ago with a customer in UK, and yes, I think it's possible. Of course, archaea, most of the time they are autotrophic, right? So it may be difficult to keep autotrophic condition in a 96 well plate. More and they grow slow. So you have to, some of them, they're not extremely slow, but some of them, they really takes ages to replicate. So I would say that for archaea, there are maybe mixotrophic, so they can use some organic compounds, and they are not extremely slow in growing. There's a possibility that this experiment can be set up, but it would certainly require some optimization.
Okay. Well, I appreciate everybody staying on. I know we are over time here, so if we did not get to your question, then we will answer separately after the webinar. Maybe I'll grab one last one here that asks about, it says, thanks for the presentation. How customizable are the FF and eco plates in terms of other functions beyond metabolism? For instance, having different carbon sources with a simple to complex structures.
So we have eco plates, they standardly made. So you can see on our website what's the list of compounds that are in there, and that's one way things can be done. Then we also provide a custom plate service. So I invite you to contact us, by email, and we'll be happy to discuss that possibility with you and see if it is possible to design a plate that is, matching your expectation and your experimental need.
All right. Well, thank you so much. And again, we'll follow up with the other questions if offline. Appreciate everybody joining us, and have a great rest of your day.
Thanks. Thank you very much, everyone. Goodbye.